Welcome to all my beloved students. Previous I have explained the basic of carbohydrates, that is the definition of carbohydrates and the structure of carbohydrates. Today I will discuss about the remaining part, that is the properties of carbohydrates, the functional properties of carbohydrates, physical and chemical properties of carbohydrates, and the uses of carbohydrates. Now, function properties of carbohydrates. As we know that carbohydrates act as a source of energy. 1 gram of carbohydrates on oxidation provide 4 kilocalorie. Carbohydrates may be used as a protein sparing actions when the diet comprises adequate amount of carbohydrates. It is used as a source of energy and the available protein is used slowly for tissue building. If the diet does not supply sufficient calories from carbohydrates, the dietary proteins tends to get oxidized and utilized as a source of energy to carry out the physiological function. Now, carbohydrates act as a source of energy for nervous systems. Glucose is the primary source of energy for central nervous systems. Prolonged hypoglycemic conditions is known to cause the irreversible damage to the brain tissue. Carbohydrate act as a carbohydrate in muscles. The major source of energy for muscular work is derived from the carbohydrates. During the process of muscle construction, the glycolysis step breaks down the glycogen to lactic acids, and further oxidation of carbon dioxide and water, and thus produced energy for carrying out the muscular work. Now, carbohydrate act as a primary source of energy for heart muscle. Glucose is used as the main source of energy in hypoglycemic conditions. A drastic changes in the functioning of the heart can be seen. Synthesis of riboses from glucose. Riboses is found in RNA. RNA means ribonucleic acids, and in nucleotides. It is synthesized in the body through a process known as hexose monophosphate pathway. And carbohydrates can be used as a storage functions. Excess calories from carbohydrates will be stored as a fat in adipose tissues. Depending on body need, the stored fats would be mobilized for energy purpose, that is the during and illness, fasting, etc. Now I will discuss the very important parts, that is the properties of carbohydrates. There is the two properties, physical and chemical properties. Now come into the physical properties of carbohydrates. First of all, absorption of waters. All you know that when a sugar or carbohydrates kept in open surface, it get moisturized, or it absorbs the moistures. So we can say that, and become sticky. So we can say that carbohydrates is a hygroscopic in nature. But some carbohydrates, some polysaccharides like starch is not soluble in cold waters. Now, solubility, as a matter of common observations, we know that sugars dissolve in water easily. The solubility of sugars in water is its uses in foods preparations and processing at room temperature. The order of solubility is as follows: fructose, then sucrose, then glucose, then maltose, then lactose. Now, mutant rotation. The optical rotation of alpha and beta forms of glucose is found to be different. While the alpha form shows a specific rotation of plus 112.2 degree. So question arises, what is rotation? So when a solution is kept in a polarimeter or plane polarized lights, it becomes rotated. This rotation may be clockwise or anti-clockwise, and this is called the specific rotations. When clockwise is plus and anti-clockwise is minus. Okay. Now, glucose have a two forms, that is the alpha and beta form. So alpha form get rotated plus 112.2 degree, and the beta form rotated plus 18.7 degree. Further, when dissolved in water, these glucose molecules, the specific rotation changes slowly until it reaches equilibrium volume of plus 52.7 degree. Now, this gradual changes of specific rotation from plus 112.2 degree or plus 18.7 degree to 52.7 degree is called the meteor rotations. It is a result of the interconversion between the two forms through the open form of glucose to an equilibrium mixtures containing 36% of alpha and 64% beta anomert. Now, inversion of sugars. This is the very, very important properties of the sugars or carbohydrates. So we know that sucrose is a disaccharide containing two units of monosaccharides. One is the glucose and fructose. Now, when sucrose is kept in a polarimeter or plane polarizer, it gets rotated, and when it rotates in clockwise, it's called the dextrorotary. When it rotates anti-clockwise, it's called the levorotary. So glucose here shows dextrorotary rotations, but fructose shows levorotary rotation, highly levorotary. That's why whole solutions it becomes levorotary. That means invert the solutions. That's why this sugars is called the invert sugars. Honey is the most uh, honey is the most uh, example of the inversion of sugars. Now taste. All the sugars are sweet in taste. Their degree of sweetness varies one another. According to the literatures, report to decreasing order of sweetness. These sugars as follows: fructose, then sucrose, then glucose, then maltose, then lactose. Now crystallizations closely relation to the property of solubility of crystallization. Crystallization is the process of formation of crystals from the solution of a substance. It has an inverse relationship with the solubility. That is, more soluble the substance, lesser is the tendency to form crystals. Crystallization also depends upon the factor like nature of the solution and addition of some other ingredients in the solutions. Effect of heat: when sugar is heated, these melt at their melting point. Above the melting points, these dehydrate, decompose, and polymerize, forming a brown mass called caramel. The process of caramel formation is called the caramelization. Toasting of bread and roasting of rice flakes are some of the example. Starch also becomes browning on heating, and its flavor changes. So, if you look that or notice when in our household we get some bread and heating it, it becomes brown colors. That means here caramelization has happened. Sorry. Now we will discuss a very important steps or properties of carbohydrates, that is the chemical properties of carbohydrates. Uh, so when we discuss about this process, we have to consider there is three reactions that happened. There is the reaction of monosaccharides, oxidation of monosaccharides, and reaction with phenylhydrazine. So reduction of monosaccharides, if we see our oxidation and reduction series, alcohol when oxidized, it becomes aldehyde, and when it oxidizes, it becomes acids. Similarly, when acid is reduces, it becomes aldehyde, and aldehyde when reduces, it becomes alcohol. So consider the glucose structures here. Basically, two functional groups. One is aldehyde groups, and another is OH groups. Aldehyde group is called the primary functional groups, and OH group also called the secondary functional groups. So when in case of reduction of monosaccharides, only primary functional groups, that is the aldehyde groups, are affected. But in case of oxidations, both primary and secondary functional groups are affected. Now, chemical properties of carbohydrates. So consider the glucose structures. Uh, consider the glucose structures. Uh, here, when glucose is treated with the hydrogen or nickel catalyst, it becomes reduces and produces sugar alcohol. That is, its two ends becomes alcohol group present. This sugar alcohol, basically, its root name is glucitol, and it extracted from a plant called sorbus and or sorbitol. So when glucose is treated with the hydrogen nickel catalyst, it produces sorbitol. This sorbitol basically used as a sugar replacement of many processed foods. Previous lectures I've discussed about the monosaccharide. It's have a two forms, that is the aldoses and ketoses. So now consider ketoses forms, that is it contain basically ketone groups. Now consider the structure of fructose, where at C2 carbon C2 carbon present here carbonyl groups. So basically we cannot create it any we cannot create any or any reactions here happened. Not it is basically impossible because it contains double bond characters. But by hybridization process, it may be possible. So it treated with the hydrogen or nickel catalyst, it produces two epimers, sorbitol and mannitol. Now, oxidation of monosaccharides. When oxidation process is done by the uses of oxidizing agent. Basically, there is two type of oxidizing agents. Another one is bromine waters or weak oxidizing agent, and another is concentrated nitric acid or strong oxidizing agent. So when treated with the glucose in bromine waters, it produces gluconic acids, and when glucose is treated with the concentrated nitric acids, it produces glycolic acids. Now, oxime formation reaction with NH2OH. An oxime is a chemical compound belonging to the imines with the general formula R1, R2, basically a large group, cetal NOH, and produces oxime. So, oxime can be synthesized by condensation of an aldehyde or a ketone group with the hydroxyamine. Oxygen formation, it is the very important properties of carbohydrates. When carbohydrate is reacted with the excess of phenylhydrazine. So, first of all, oxygen is a class of carbohydrates, derivative found in organic chemistry, formed when sugar are reacted with the phenylhydrazine. The famous scientist Fischer developed and uses the reaction to identify sugar whose stereochemistry differs by only one chiral carbon. So, question is, what is chiral carbon? Basically, chiral carbon is the carbon whose have four functional different groups. This carbon called the chiral carbon. The reaction involves formation of a pair of phenyl hydrogen functionalities concomitant with the oxidation of the hydroxymethane group adjacent to the formal center. The reaction can be used to identify monosaccharides. It involves two reactions. Firstly, glucose with phenylhydrazine gives us glucophenylhydrazone by elimination of waters. This glucophenylhydrazone, when treated with the uh, excess amount of phenylhydrazine, it produces oxygen. Now, RAB degradation. Basically, RAB degradation means the carbon degradations. When we consider the hexose compound, basically hexose, it has a 6 carbon. So when we created a degradations, it becomes pentoses, because pentoses means 5 carbons, then tetroses, then trioses. So reduces of the number of carbon. If we consider the like Hofmann bromide degradations, that is carbon degradation process here happened. Now consider structures where CN aldoses, it has 4 carbon group. So it treated with the bromine in waters, it produces CN-1 aldoses. That means here carbonate reduces. That means here RAB degradation process here happened. Now, caramelization reaction of sugars. Caramelization is the browning of sugars. The process used extensively cooking for the resulting nutty flavor and brown color. As the process occurs, volatile chemical and released produces the characteristic caramel flavors. Like the Maillard reactions, caramelization is one type of non-enzymatic browning reactions. However, unlike the Maillard reactions, caramelization is pyrolysis as opposed to reaction with the amino acids. All you are requested, please see in your books what is the enzymatic browning reaction and what is the non-enzymatic browning reactions. Okay. Now, when caramelization involves the disaccharide sucrose, it is broken down into the monosaccharides, fructose and glucose. The caramelization reactions are also sensitive to the chemical environment by controlling the level of acidity, the reaction rate, and the rate of caramelization is generally lowest at near neutral acidity, that is pH equals to 7, and accelerate under the both acidic and basic condition. Now, reducing and non-reducing sugars. Basically, reducing sugar is the sugar. It has 3 aldehyde ketone groups. The saccharide which reduces failing solutions, bending solution, and tolerance reagents are called the reducing sugars. Basically, these solutions are used to identify the reducing sugars. Reducing sugars contain alpha hydroxyaldehyde or alpha hydroxyketone groups. Cyclic hemiacetyl or cyclic hemiketyl groups is equilibrium with the open chain from having the free aldehyde or ketone groups. All monosaccharides like glucose, fructose, and disaccharides, maltose and lactose is the example of reducing sugars. Now, non-reducing sugars, the saccharide which they do not contain any free aldehyde or ketone groups are called the non-reducing sugars, and these sugars not reacted with the filling solution, bending solution, and tolerance reagents. And cyclic form of non-reducing sugars cannot be opened up in an open chain having free carbonyl groups. All polysaccharides are non-reducing sugars like starch, cellulose, and one of the most important example is the sucrose, is a non-reducing sugars. Now, estimation of reducing non-reducing sugars, basically the famous scientist Lenian scientist has very sincerely estimated the amount of reducing non-reducing sugars from samples and by the practitioner method using of failing solution, failing A and failing B, which is done in your lab. So please carefully notice in your lab. Now starch. Starch is a polysaccharides and it contains a large number of glucose units joined together by glycosidic bonds. This polysaccharide is produced by all green plants as an energy source. It is the most common carbohydrate in the human diet, is contained in the large amount in such staple foods like potato, wheat, maize, rice and cassava, etc. Pure starch is a white, tasteless and odorless powder, and it is insoluble in cold water or alcohol. It consists two type of molecules. One is the linear or helical amylose and branches amylopectins. Depending on the plant, starch generally contain 20 to 25% amylose and 75 to 80% of amylopectin. Glycogen, the glucose stores of animals, is a more branches version of amylopectin. The released starch consists of two fractions. About 20% is a water-soluble material called amylose. Molecular amylose are linear chains of several thousand glucose units joined by alpha C1 to C4 glycosidic bonds. Amylose solutions are also the actual dispersion of hydrogen helical micelle. And the another is amylopectin, which contain a branch network C1 to C4 and C1 to C6 glycosidic bonds and are essentially water insoluble. Now, starch dehydration process. Starch becomes soluble in water when heated. The granular starch and burst. The semicrystalline structure is lost and the smaller amylose molecules start leaching out of the granule, forming a network with the whole water and increasing the mixture viscosity. This process is called starch dehydration process. Factor depending on the dehydration process. The dehydration temperature of starch depending upon the plant type and the amount of water present, pH type and the concentration of salt, sugar, fat and proteins in the recipe, as well as the derivation technology used. Some type of unmodified native starch started swelling at 55 degrees temperatures. The detonation temperature also depends on the amount of damaged starch. Granules, this will swell faster. Damaged starch can be produced, for example, during the wheat milling process on when drying the starch cakes in the starch plant. Retrogradation, the process of reassociation of the starch granule on cooling of the gelatinous starch or the starch paste is called retrogradation process. It depends on the relative properties of amylose and amylopectins in starch, as linear amylose molecular reassociate faster than the highly branched amylopectins. Reacting starch foods also influence this process. The digestibility of starch in the small incision reduces by the degree of processing and retrogradations. If starch is subjected to dry heat, it breaks down to form dextrins, also called pyrodextrins. In the context, this breakdown process is known as dextrinization. Starch can be hydrolyzed into smaller carbohydrates by acids, various enzymes, or a combination of both the two. The resulting fragments are known as dextrins. The extent of conversion is typically quantified by dextrose equivalent, which is roughly the fraction of the glycosidic bonds in starch that have been broken. These starch sugars are by far the most common starch-based foods ingredients and are used as sweetener in many food companies. Now, food application of starch polysaccharides. Thickener, you know that starch is absorb water and swell up. If the amylose content of the starch is more, its long water soluble chain increases the viscosity and thickener. Where sausages company or padding, this company used the starch as a thickener agents. Fat replacer, the water absorption property of starch is also be responsible for the mouthfeel of many food products containing them and may be used as food fat substitutes like salad dressing and dairy products. Water binding, as starch readily absorbs moisture, it checks its escape from the food products and in some dishes like cakes, it retains the moisture and yet does not allow to be wet. The high water binding ability of starch leading to their swelling up can provide body and texture to the foodstuffs. So thank you all of you. My all the notes already uploaded in your council website, so please visit there and download it. Okay, thank you everyone.